Good day, global citizens. Welcome to another episode of Dr. Fajembola'S Opinion. On today's episode, I have a senior colleague and expert in the field of HIV and women's reproductive health. And I'm glad to bring her on board to discuss and to break down what we should know with regards to HIV and a specific species of rat known as the Wister rat. So join me to welcome Dr. Bakare Airat Adeola. Welcome on board, Ma.
Thank you very much, Doc. Happy
Thank
to
you
be
so much.
here.
Thank you
Yeah.
so much. Thank you so much for honoring the invite and choosing to share your knowledge and your time with us. So, first of all, as I said earlier, we are discussing what we are discussing today is the disruption of the um, female reproductive system in terms of hormone at the hormonal level. Specifically, we're looking at the Wister rat and the effect of antiretroviral regimens on this rat. So, first of all, let's give our audience an overview of Dr. Bakare's biography. She's a lecturer and translational researcher at the University of Lagos, Nigeria, who is interested in investigations that impact directly on lives. She holds a Ph.D. in anatomy and specific focus on reproductive biology and neuroendocrine regulations. Our professional journey reflects commitment to advancing scientific knowledge and improving women's reproductive health through evidence scientific explorations. Currently, she's interested in unraveling the mechanism behind neuroendocrine dysregulations in antiretroviral drugs associated infertility for strategic development of treatment intervention. Dr. Bakare has received several competitive research grants, presented her research findings at both national and international conferences, and published in reputable peer-reviewed journals. Beyond academia, she's passionate about mentorship and community engagement. She actively supports teenage girls through empowerment programs focused on sex education and life building skills, thus fostering informed choices and resilience in young girls in building greater tomorrow. Dr. Bakare's career combines scientific excellence, teaching and social impact, positioning her as a scholar dedicated to improving reproductive health, enhancing knowledge acquisition for continuous growth, and shaping future generations of leaders. Dr. Bakare, I got tweets as I usually do to see what global citizens are saying about HIV in women in regards to their reproductive health. The first I got was from Kechuku Bona, where he said the AIDS Healthcare Foundation, AHF Nigeria, has raised the alarm over the rising rate of new HIV infections among young women, revealing that about 4,000 women aged 15 to 24 become newly infected with HIV every week, with over 3,300 of those cases occurring in sub-Saharan Africa. The second is from the Institute of Global Health, where they awarded Dr. Chemtai Mungu with $2.5 million to improve HPV treatment outcomes in women living with HIV in Africa. That's good. Seeing more investment in the reproductive health of Black women. The last tweet is from Dr. Winnie Banyima. She's a loud voice for HIV in Africa, even globally. She's the director, I guess she's the director general of UNAIDS, where she said, "My warmest congratulations to Qureisha Abdul Karim on receiving the Virtual Prize 2025. I am inspired to see your pioneering contributions in understanding the HIV epidemic among young women and your long-time advocacy for the rights of people living with and affected by HIV duly rewarded." So, Dr. Bakare, let's dive deeply into that discussion now. um, antiretroviral therapy. I know I'm a medic, and I know of course I know what antiretroviral therapy is, but we want this to be what a layman can understand, a civil servant in accounting, someone flying a car by the road. So what is the difference? And I put in a way that they can understand the different terminologies. So, what is the difference between antiretroviral therapy and highly active antiretroviral therapy?
Yeah, I'll try as much as possible to, break it down to a layman understanding. Antiretroviral drugs generally are drugs that help to, suppress viral growth. They help to reduce the replication of the virus, because that is the main, will I say, essence of survival of the virus, which we now see as HIV. So it helps to suppress its replication. But for highly active antiretroviral drugs or therapy, basically, is a combination of specific drugs, that have been tested to be most effective. What they do basically is to combine two nucleotides and one non-nucleotides. So they are, I don't really want to bring these big words, because I don't know your audience. We need to break it down to the layman understanding. So basically, highly active antiretroviral therapy is also a combination of antiretroviral drugs, though, but they just select like between 3 to 4 of them, combine them together to get the best effect. So that is what it is basically. It is still a collection of antiretroviral drugs, but for the highly, in quotes, something that is highly should mean that the capacity is more enhanced. So they try to combine a number of drugs that work through different mechanisms, why one is helping to, reduce the genetic replication, the other one is helping to alter the shape so as not to allow, replication to occur. But I don't want to go into the mechanism of how they act. But basically, highly active antiretroviral therapy is a collection of 2 to 4 drugs, that have been shown to be most effective. And this is what they use for patients that are infected, and in helping them to suppress, the growth of the virus, which of course is the target. If they are suppressed, the viral load will be reduced, and the immune will boost up, and things will begin. Other reproductive and other systems in the body will begin to, assume their functions.
Thank you so much, Dr. Bakare, for that beautiful explanation. You gave a very simple distinction of ART from HAART. So, Ma, please, what are 3 things we should know about highly active antiretroviral therapy on the reproductive system?
Yeah. All thanks to antiretroviral therapy. We know that without it, maybe the world would have been eroded by now. But it's something that has come to, transform HIV management to a chronic disease condition to what can be managed. You see people with HIV now, they live their normal life, all thanks to antiretroviral drugs. But however, there is a current emergence of its effect on reproductive health. There are a number of research, if you go through literature, that are popping up, particularly focusing on the long-term, usage of this therapy. Like in my lab, we use both human and also experimental models, because whatever we do with the experimental models, specifically the Wistar or the Sprague-Dawley rats, be translated into human. So, extent that we can work on human, we try to do, while the rat will provide other, specific target for us. So in our lab, we've, discovered that this antiretroviral therapy, which of course we say is a combination of drugs, tends to disrupt the endocrine profile. It tends to affect neural endocrinology, and we know that most virtually all the systems in the body are controlled by the brain. And if this drug is altering the neuroendocrine pathway, it is going to go down to the gonadal level and disrupt reproductive function. So I would want to say that a number of studies now are popping up, confirming that these drugs truly, they disrupt the endocrine system and also creating infertility indices. We worked on human and we discovered, menstrual irregularities in HIV positive women that are on these drugs. Where we feel that it is this drug that is the problem is that we try to compare these menstrual irregularities also with women that are positive but are naive to these drugs, which means that they are not, placed on it. We see that we have more of menstrual irregularities in the women that are on this drug than those that are even positive and are not on it. So that is what actually drove our interest to focusing on the drugs. So we decided to go into the lab, where we can really target on the drugs, because to, infect Wister rats with HIV is not something that can be easily done. So we decided that we are going to be administering the drugs while we check along the reproductive axis, the effect that it has. And we've also came out with a number of, findings that have also been published. So we see that even in experimental models that are uninfected, these drugs have been able to disrupt the hormonal profile, Even at the ovarian level, they've been seen to, disrupt folliculogenesis. So we have a number of studies in literature, both the ones also that we carried out in our lab, and we've seen that there is an emerging crisis that is needed to be looked into.
Thank you so much, Ma, for that. At the beginning, we mentioned the Wister rats, and it's my first time actually hearing about them. I know I've heard of other species of rats, and this is why, because when we do this, people think, oh, it's only for them, the audience. Actually, it's also for people who have not heard some terminologies before. I've heard of the ART, ART, but the Wister rats. And it was repeated severally in the paper. What is unique about these rats?
Yeah, Wister rats are experimental models that are used in basic laboratory research. We know that basic laboratory research basically is like the root. That's why they call it basic, From there, it will translate into clinical application, clinical trial before you have final discovery or final interventions made. So it starts from the basic. And if you are doing something from the basic, there are regions that are unethically, they are not approved ethically for you to assess them in human. For example, the brain. You cannot say you want to do a research on human. I cannot crack up your skull and start investigating on your brain, but I can do that in, Wister rats. So they try to extend the limit at which we basic researchers can go, because they create that platform that it is not allowed in human, you can do in rat. Most especially is the fact that the Wister rats are very easy to handle. You can create a small cage, put them inside, monitor them. They fit an experimental environment. Then another key thing why Wister rats are also very good for basic research generally is that they are mammals. When you try to classify them, they are also mammals. So the physiology of the human body can be replicated in them. So whatever areas that you are studying, you can also study in them, and you can use it to assume or predict whatever that will be happening in human. Apart from Wister rats, you can use albino, you can use Spragdali. I think I even work more with Spragdali. So they have different species because they are also mammals. Whatever you have, they also have it. It may not be as large. Their organs, they are small too. So, and that is why they are easy to handle and also manipulate. And they are very easy to work with. But focusing on reproductive research that I am, specialized in, for reproductive research, the reproductive hormone profile perfectly, aligns with what mimics human. So it's even more interesting for me to work with them. whatever that I get at the end of the day, I can easily take it to the human study, which is the advanced one, and be able to interpret whatever I have there for my continuous advanced reproductive quest. So that is what we use basically for basic laboratory research. And if you have a very good ethical, body in your institution, before you also work with them, I know clinicians work more on human. We do ethical approval when we are working on human, because I also collaborate with doctors too in other institutions. As you do for human too, you respect them because they are also living. So you also seek ethical approval, whatever things that is needed, the requirements, the protocols too that you want to use on them. Because at the end, you may be sacrificing them. You cannot just come and put knife on their neck. No, we have standards. For people that are into basic laboratory research, they know all this. So it is not really an easy thing too. So we won't say because they are rats, you can handle them anyhow. You also need to handle them with respect, just as what you would do to humans.
Okay, thank you so much. Thank you so much, Ma, for updating our knowledge on the Wistar atom. So anyone who is listening and who is watching this now understand what the Wistar is. These words were repeated in your paper, replacement, reduction, refinement. Please, can you shed more light on the three R's in animal research?
Yeah, in animal research, the three R's basically is replacement, reduction, and refinement. For replacement, we know that for experimental research, especially for basic laboratory scientists, there could be other alternatives. If there are alternatives that one can use, because it's going to also be helping to limit the wastage of these animals. This is what we use, and we cannot afford to just, allow them to go into extinction due to the way we use them. So if there are alternatives, you can replace them. Some people work on computerized virtual model of cells. They create it on, and they work on it. They use it to make interpretations and also deduction to offer solutions to problems. You can also use cell lines. I have students that have worked under my supervision that have worked on cell lines. So you can get specific cell lines of your target, You can also use them for experimentation. That will also help in, replace whatever you have in terms of animal research. And for reduction, before we got into this space, the last topic we discussed on, I mentioned something about ethical approval. I think one of the aim of ethical approval committee is for them to limit or reduce the wastage of these resources that we have. So you should be able to quantify appropriately, because there are actually models or formulas that you can use to derive your population size, so that you won't just be working and wasting resources, trying to protect the animals, species too. So helping to reduce them unwanted, harm to their lives. Then also refinement. There are procedures too that would need to go through ethical approval, like the one I said at the end of the research. Some scientists may want to sacrifice. Maybe you want to extract organs. If you are extracting organs, you cannot extract the organs while the animals are alive. You need to sacrifice them. So you need to do or employ protocols that are, ethically guided, so as to minimize pain, suffering, or distress. All these things are things that animal research look into, and they prioritize them for continuous, application.
Thank you so much, Ma, for that, for explaining the distinction between replacement, reduction, and refinement. Also, we are we are understanding the terminologies so that anyone who listens to this already understands what the paper is about, or wants to go back and read on the paper, have more understanding on what the paper is about. You mentioned the anti-Mullerian hormone, follicle stimulating hormone, luteinizing hormone, and estradiol. These are hormones present in humans, and as you mentioned, the Wistar rats are also mammals. What is it about these hormones in regards to our reproductive system?
Yeah, we know that the reproductive system generally are controlled by tightly regulated hormone profile. Why I use the word tightly is that there are levels that which they must be before some reproductive activities must occur. And that is the peculiarity with the female reproductive system. It is very, very distinct. There's a way God has created it, and that is why we are unique, though. So all these hormones, we need to, come together. There is actually an interplay between them, where one will, impact on the other to be released. This will impact on the other to be released, then it will impact on the organs. I don't want to go into details because we are not in an anatomy class and I don't want to take the whole of the time. So I'll just summarize, I'll try and go to the root, which is the hypothalamus. The hypothalamus is a sub-region at the base of the brain. This is where reproductive effects are geared from. If you want to start your car, that is where you start that car from. So from the hypothalamus, we have what we call the Kixpeptin. The Kixpeptin hormone is what, will be released at that region to stimulate the release of gonadotropin-releasing hormone. The gonadotropin-releasing hormone also in the hypothalamus will then stimulate another gland that is just close to it, which is the pituitary gland, to stimulate gonadotropins, which are basically the follicle-stimulating hormone and luteinizing hormone. So the pituitary secretion of gonadotropin from that brain region is targeting the ovary, which is located in the pelvic space. And this is where it stimulates folliculogenesis and luteinization. So you can see how this network is from the brain, driving it down to the pelvis, where it is now exerting effect on the ovary and also the uterus. And basically, these are to, enhance finally carrying a baby. Or let me start with ovulation, implantation, growth, then finally delivery of the baby. So that is how they work. But for anti-Mullerian hormone, it is produced by the granulosa cell of the ovary. And what it does is that it helps to control how folliculogenesis occurs. Don't forget that in the ovary, that is where the egg is. The egg is not just, lying naked in the ovary. It is covered by groups of cells that we call the granulosa cells. These are like the protective coat or gadget that it wears. It is not only protecting it, it is also helping to carry or deliver nutrients to the egg that is lying within it. So this granulosa cell produces anti-Mullerian hormone, which of course will help control how follicles are growing. Don't also forget that within the ovary, the follicles are growing, a number of them are growing, but it is just one of them that would advance to the final stage. So in order to prevent pools or excess follicles from growing, the anti-Mullerian hormone will help to control limits that, look, let's just take 10 of you, begin to proceed in this journey of folliculogenesis. The final one is just one that we need at the end of the day. So the final one will be released. The other 9 will be attractive, and it's going to be wastage. And we know that women are born with these follicles, they are not replaced. So so in a way, it's helping to gauge how those, follicles will begin to resume growth, in helping to control it. So that is why they say that if you measure the level of anti-Mullerian hormone, you can use it to depict your ovarian reserve, because this is what actually controls, how the follicles drive into growth, why it allows some of them to just remain at that lying stage. So that is what the anti-Mullerian hormone does. Then for the follicle stimulating hormone, if you drive back to the previous explanation, I said that the follicle stimulating hormone is produced by the pituitary gland. Specifically, it is the anterior lobe that produces follicle stimulating hormone. And it does not just wake up to secrete this hormone, it is stimulated by the gonadotropin-releasing hormone by the hypothalamus. So for this FSH, from the name, when I tell my students, I don't know if some of my students will be opportune to watch this video. Follicle stimulating hormone. The name already tells you what it does, It is stimulating ovarian follicle. That is what it does. So it targets the ovary, because it is in the ovary that you have ovarian follicles. So it begins to disrupt their development. They begin to, proceed from the primary stage to the secondary, to the tertiary, to the Graafian, before they finally get ovulated. So the specific role of follicle stimulating hormone is to stimulate folliculogenesis, which of course is the growth of the ovarian follicle. While the luteinizing hormone is also produced by the pituitary gland, but what it does is to stimulate or to exert effects on release of an egg. If you go back to how hormone provides her, the surge in LH is what will, cause the release of the hormone. And after the release, the release of the egg. And after the release of the egg, you have a shaft that we call the corpus luteum, which of course will either be transformed to corpus luteum of menstruation or corpus luteum of pregnancy. So it is the luteinizing hormone that also enhances this development. Then the last hormone that we have is the estrogen, which are the steroids. It's produced by the ovarian follicle. Then we have the progesterone.
Yes.
Yeah. So for progesterone is the pregnancy hormone that helps to, prepare the uterine wall for implantation and also sustain the growth or the development of the implants. So this hormone generally, you see that they are released from different ends, but they work synergetically together. One will enhance the release of the other, all in the quest of, enhancing reproductive function.
Thank you so much, Doctor, for that. You've gone even beyond the question. Thank you so much for that. For
I'm
allowing
trying
our
not to do too much. I'm just trying.
It's perfect, ma. It's perfect, ma. You're giving the explanation we need to understand the female reproductive system and how fertility works, which I feel it's a knowledge every adult, a man or woman, should know. And thank you so much for breaking it down the way we want it. Please, Dr. Bakare, infertility, you mentioned subfertility in the paper, and I feel our audience would like to know the difference between infertility and subfertility. Are they the same? Are they different?
Yeah. Yeah, I would say they are almost the same, but they are specifically different. Infertility basically is the inability to conceive after maybe a year of regular sexual intercourse and thereabout. But for subfertility, from the name, as I said, also sub, it means something that is reduced, like it takes longer to occur. Subfertility, somebody who experiences this may eventually get pregnant, but not at due day, as when you expect. A very good illustration that I would want to use for this is maybe someone that just got married. And after marriage, generally it is expected that you get pregnant. It does
Yes.
not occur immediately. A lot of factors can be factored in. Maybe there could be obstructed uterine tube, there could be ovarian irregularities, a lot of things can occur like that. So when it does not happen at when due, but infertility basically is the inability for it to occur. And of course, there are also factors that can be, attributed to it, which could be maybe sexually transmitted diseases, it could be genetic, and a lot of other intense factors can be responsible for that.
Thank you so much, Ma, for distinguishing subfertility and infertility. I believe with this, global citizens won't use the words anyhow. I know the difference between a subfertile human and an infertile human. Ma, please can from the paper I got that there is a marginal decline in the ovarian reserve of females above the age of 30 years and a significant decline in the ovarian reserve above the age of 40 years. What else should we know about the ovarian reserve? What are things we should know about the ovarian reserve?
Yeah, ovarian reserve basically depicts the quality and the quantity of egg that the ovary has. The pattern by which folliculogenesis, occur is very distinct. I still cannot figure out any other, processes in the body that is like folliculogenesis. It's just distinct, So when it occurs, there is a particular number that we are born with as women. And as folliculogenesis occurs, it's a monthly event, like a menstrual cycle. So as it occurs, a specific number also will go into the pool of development. And as they go, we say only one of these follicle that has the egg will assume the final stage of getting ovulated. And after ovulation, we know it's going to be implanted. It's going to be fertilized before implantation will occur. Then you now feel this pregnancy has been achieved. The remaining one will become atretic. So it's more like a wastage. And this is not going to be replaced. So as you begin to take the pills every month, there is a gradual reduction, So naturally, it is going to be reduced. But there are also other factors that can, further disrupt or reduce this number, Like the anti-Mullerian hormone, we say we can use to measure the level of this, the quantity of egg that we still have. And this is because it helps to control. So if there is a dysregulation of the anti-Mullerian hormone that is already helping to control it, okay, maybe it's just 10 that should go into recruitment, that should be recruited to proceed for growth. The anti-Mullerian hormone is not exerting its effects at optimal, gauge. It is now taking like maybe 50. Okay, the 50 of you can go. And you know that this 49 will be wasted. I hope you get it.
I understand.
Sorry. So 49 will be wasted. So the control of the anti-Mullerian hormone is also very, very important. So you see that naturally on its own, there is a reduction. And it has to reduce. It's logical and understandable because it's a process that cannot be replaced. And they're already going into that process and you get them hurt along the way because one is already, exceeding it. And that is the one that will finally be ovulated, while the other ones will just remain atretic and they cannot be replaced. And this is where the problem is. If they are going to be replaced, there's not going to be problem. Just one will be moving and you have pools of them.
Yes.
So it's a lot. So you see that, and considering the kind of exposure that we have now, a lot of things, a lot of factors, lifestyle and all other things too will further impact negatively. Exposure to, radiation, smoking, also certain drugs too, can help to, disrupt this organization, Studies have also shown that antiretroviral drugs also can disrupt the control of this anti-Mullerian hormone. So helping to, at the end, reduce the ovarian reserve.
Thank you so much, Ma, for shedding more light on the ovarian reserve. I believe every man and woman listening to this, and I'm not concentrating on the woman race. I believe every man and woman listening to this has more knowledge on the ovarian reserve and what factors affect its number. We are what are they calling it? We, from the paper, ARD was discussed, and from the paper I quote, which ARD has the highest potency on the female reproductive system. The antiretroviral drug, ARD, antiretroviral drug. I should not use terminologies. A few were mentioned, the favirenz, tenofovir, and the rest. Which ARD has the highest potency on the reproductive system?
Well, currently in our lab, we are combining, although we got a grant recently that will now, start to really check which of the particular one. We are currently carrying out a study on that. But from literature, there are 3 that have been established, to disrupt greatly the affairs of the female reproductive system. And these are the liponavir, ritonavir, and indinavir. Those 3 have been established. But in our lab, we are currently working to see, because these are not even in use anymore, to be honest. The ones that are currently being used, we are, researching on them to check which specifically, like the other studies that we didn't really, go into details of checking which specifically, but we saw disruption in a number of them. But now we are targeting just individual drugs to see, which of them in particular is causing this effect, especially at the neuroendocrine level, which is the brain axis. So this is what we are doing presently.
Thank you so much, Ma, for that. We'll be keep encouraging you, and we'll pray you get more funding to do more research on this. The last question, which I usually ask most of the researchers in relation to their publication, on the fertility spectrum, and where do you see women on highly active antiretroviral therapy in the next decade?
Well, in the next decade, I don't want to say not too good, but I'm hopeful for a successful intervention, especially as it relates to reproductive health. And this is as a result of what we are currently seeing in our labs now, and also in literature, the effect or impact that highly active antiretroviral therapy, is projecting on reproductive health, especially targeting females, because we work on female. I know some other lab may be working on male, which of course will now give a comprehensive understanding. So with what we are seeing now, and the essence of research now is for us to be able to offer solution, because research will make you understand the details of why these problems are occurring, the basis behind them. And when you understand the problem, definitely you'll be able to, offer solution to that. So we are hoping that at the end, after carrying out this research, we would be able to develop an effective intervention, maybe through drugs or drug modification. We are targeting some hormone replacement therapy, though, but we've not gotten there, but that is our future aim. So at the end, if we put all this in place, I want to believe that it's going to be, a success at the end. While the HIV positive women or couples are fighting already with their status, their reproductive health also will also be protected. And I would want to say that with adequate funding, I was happy that, you mentioned it previously, funding is also very essential. For you to carry out the scientific research is not a small deal. Even the kits alone are millions now. If you look at the dollar rate, some funds, when you apply for grants, by the time they release the grant, the expenses are already doubled up. So research funding are very essential. If you have adequate funding, you'll be able to execute your research effectively. And the aim of that research, of course, will be, achieved at the end of the day.
Thank you so much, Ma, for having a beautiful discussion, for giving us a beautiful discussion. Let me put it that way, because I cannot do it alone. I need guest researchers who are experts in the field to break down these terminologies and also explain what is happening in the research world pertaining what they are working on. So thank you so much for spending your time with us today. And we looked at Global Citizens. We looked at Dr. Bakare's publication during the week. If you've not read it, please go through it, read it. It's quite explanatory. It's quite easy to understand. And there is hope. One thing is, aside improving the well-being of global citizens, on papers, this is not the first time we're looking at HIV. On papers like on HIV, publication on HIV, it shows that there is hope. The life expectancy of people living with HIV has increased. You can be positive and you have HIV negative children. So there is hope for you who you have HIV. And also through platforms like this, discussions like this, we are trying to also reduce the discrimination, the stigma associated with HIV and AIDS. So please read the paper and discuss with colleagues, and also share our discussion. Thank you so much for joining us, and have a blessed week.